Good morning. Good morning. Very warm greetings to all dignitaries, guests, and delegates. With great joy and immense exultation, I, Dr. Charu Chaturvedi, Assistant Professor Botany, DAV PG College Kanpur, is hosting the event today and feeling privileged to extend my warm welcome to all of you present here today for Dr. Nagesh Swarup Memorial Lecture Series 2022, sponsored by DST SERV under Accelerate Vigyan Vritika Scheme in collaboration with Indian Science Congress Association, Kanpur chapter. We are excited to announce more than 200 registrations and large number of participants are joined with us from across the globe. The curiosity to know and learn more and more is the ultimate key to growth and success. So to increase the parameters of our knowledge, we started this program of lecture series since pandemic year 2021, which was officially the year of virtual and hybrid events. Now it is successfully reaching its goal. Today we all are here to comprehend important and current scientific inventions and beneficial information about the vast topic like stress adaptation in crop plant and small are big. Yes, small are big nano potentials to enhance the plant resistance against stress. Now we would like to request all dignitaries for lighting of the lamp before Ma Saraswati. Sir, please.
म्यूट कर दे मुझे।
जैसा कि हम जानते हैं हमारे प्रेरणा स्रोत नागेश स्वरूप जी जो कि आज हमारे बीच नहीं रहे परंतु उनके आशीर्वाद से ही आज हम यहाँ तक पहुंचे हैं। हम सभी उन्हें स्मरण कर सादर प्रणाम करते हैं। सादर नमन है सर आपको। हम सभी सादर नमन करते हैं। कार्यक्रमों की श्रृंखला को और आगे बढ़ाते हुए मैं वृत्तिका स्कीम के इंटर्न मिस्टर शिवम को बुलाना चाहूंगी, जो कि यहाँ आकर हमारे परम आदरणीय प्रिंसिपल सर को सम्मान स्वरूप बुके प्रदान करें। शिवम। प्रिंसिपल सर को। सर, स्वागत है आपका। अब मैं पुनः वृत्तिका स्कीम के इंटर्न मिस तीक्ष्णा को बुलाती हूँ, वे आए और हमारे गेस्ट स्पीकर डॉक्टर विजय प्रताप सिंह सर को पुष्प गुच्छ प्रदान करके सम्मानित करें। स्वागत है सर आपका। इसी कड़ी को आगे बढ़ाते हुए मैं मिस काजल वर्मा, जो ये भी वृत्तिका स्कीम के इंटर्न है, को यहाँ बुलाऊंगी, जो डॉक्टर सुधीर कुमार श्रीवास्तव सर को आदर स्वरूप बुके भेंट करें। अब मैं मिस्टर वैभव को बुलाऊंगी कि वे यहाँ आकर एचओडी सर को सम्मानित करें। Now, I will heartily welcome the principal of our college, Dr. Arun Kumar Dixit sir, for the welcome address. Welcome you, sir.
Very good morning to all of you, my colleagues and my dear students. As the principal of this glorious Heritage College, I truly feel honored to welcome you all to this commemorative lecture series being organized in hybrid mode in fond memory of our former Secretary, Board of Management, DAV College Kanpur, Dr. Nagen Swaroop ji. It is the part of our regular academic engagements organized by various departments of the college as Dr. Nagen Swaroop Memorial Lecture Series. It is being organized under the aegis of Indian Science Congress Association, Kanpur chapter, and sponsorship of DST SERB. I am feeling overwhelmed by the gracious presence of great luminaries from the field of academics on this auspicious occasion. I convey my warm greetings to our Secretary Board of Management, Srimati Kumkum Swaroop ji, who keeps inspiring us all for escalated academic engagements. I convey my sincere greetings to young and dynamic engineer Gaurav Swaroop ji, who is inspirational as the Joint Secretary of the Board of Management of the college. I express my gratitude and greetings to other respectable dignitaries who are gracing this occasion, like Dr. Vijay Lakshmi Saxena, President ISKA, and Dr. Sudhir Srivastav, Treasurer, ISKA Kanpur chapter. Today's guest speaker, Dr. Vijay Pratap Singh, I also greet you on this occasion on behalf of DAV College Kanpur. I wish all the best to the organizers and the participants. The topic Stress adaptation in crop plants is of great relevance for India as a country of extensive resource and climatic diversity. Agriculture is the mainstay of the major chunk of India's population. Improvement in purchasing power of farmers provides huge linkages for industrial and services demands, accelerating the overall economic performance of the country. Agronomy in India has had a checkered past, but with introduction of technology and use of improved varieties of various crops, we have transformed from our status as net importers to net exporters in food grains. Stresses are the factors which interfere with potential scale of yield. Engineering tolerance to various stresses in high-yielding cultivars has direct bearing on quantum of productivity. The experts will shed more light on technical aspects of the topic, and I stop here to allow them to take over the dais for a detailed light on the topic. Thank you all for a patient audience to my views. I wish a grand success to organizers in terms of a scale of output of the lecture series. Jai Hind, Jai Bharat.
Thank you so much, sir. It is my proud privilege and honor to welcome Dr. Vijay Lakshmi Saxena, ma'am, President, Indian Science Congress Association. Thank you, sir. Dr. Vijay Lakshmi Saxena, ma'am, President, Indian Science Congress Association, for inaugural address. Ma'am is online with us. You're welcome, ma'am. Namaskar, ma'am. Most welcome, ma'am. It is my proud privilege and honor to welcome Dr. Vijay Lakshmi Saxena, ma'am, President, Indian Science Congress Association, for inaugural address. Ma'am, please guide us. You're welcome, ma'am.
Ma'am, your voice is not proper. Voice, no voice. Please unmute. मैम की वॉइस नहीं उधर से है मैम की वॉइस उधर से नहीं है आवाज आ रही कुछ मैम की आवाज उधर से ही ऑफ है मेरे पास
और अनम्यूट करो कह रहे हैं। अब आने लगी कह रहे हैं।
Yes, voice is clear, ma'am.
अब आ रही है।
Okay, thank you. Okay, but I could not be able to hear your voice now, so that's why I asked if I'm audible. Yeah. Should I start now?
Yes, ma'am.
Okay. A very good morning to you all. Present here for a very important lecture on stress adaptations in crop plants. Mrs. Kunti, and Dr. Ashok Saxena, Past General President, Dr. Manoj Chakravarti, Past General President, Dr. Arvind Saxena, General President-elect, Dr. Ramakrishnan, General Secretary, Membership Affairs, Dr. Anup Jain, General Secretary, Scientific Activities, Dr. C.O. Sarprakash, Treasurer, Indian Science Congress, Dr. Niveta Chakravarti, EC Member, Dr. A.K. Dikshit, Principal DAV College, both the local secretaries, Professor Rajesh Singh and Professor Khadekar, Professor Govardhan Khadekar, Dr. Tripathi, Speaker, Dr. V.P. Singh, Speaker, Dr. Sudhir Srivastava, Treasurer, ISKA Kanpur Chapter, Dr. S.P. Singh, organizer of the event, organizer of the today's event, Dr. Atul Kumar, Executive Secretary ISKA, and Dr. Devashri Datta Saha, Hindi Officer, Indian Science Congress, Kolkata. Really, so many people, they all are attending your lecture series. Really, I am so happy today. And I am very glad that ISKA Kanpur chapter is in association with DAV College Kanpur is organizing Dr. Nagen Swarup Memorial Lecture 2022 on stress adaptations in crop plants. All the participants of the program, guest speakers, faculty members, and students from DAV College and from other institute and colleges, my beloved student fraternity, media personnel, ladies and gentlemen. I am glad that ISKA Kanpur chapter, in association with the DAV College Kanpur, has organized Dr. Nagen Swarup Memorial Lecture Series 2022. First of all, I feel privileged to have been granted the opportunity to express my views on today's lecture. I wish to thank Dr. Sudhir Srivastava and Principal of the College for inviting me to share my views with you all. Today, I am not going to discuss anything about the stress adaptation in crop plants, as my another meeting is going on, and I will take just few seconds because the meeting is going on here at RTM University Nagpur, where our 108 Science Congress is going to be held from 3rd to 7th January 2023. I would like to talk few words with a famous quote of Rabindranath Tagore: the highest education is that which does not merely give us information, but makes our life in harmony with all existence. I strongly believe that Tagore's idea of imparting true education had the inherent seed of sustainability much before the term sustainable development was officially coined in late 20th century. As you all know, the harmonization of society's development with our planet's environment is the basic foundation of sustainable development. It is our duty to spread the concept of sustainable development nationally by pointing out the interdependence of scientific research, production, and environmental limits. Today, I'm proud that the DAV College, in association with the ISKA Kanpur chapter, has organized this lecture series. And I'm not going to discuss anything about the theme of about the lecture, topic of the lecture, and you please all go ahead with your lecture series. I know that very eminent speakers are there, and wish you all success for the conduction of this lecture series. Thank you all. Thank you very much. Excuse me, should I leave the meeting? Because another meeting is going on. Thank you, Dr. Charu Chaturvedi.
Thank you so much, ma'am. Thank you so much.
Okay.
Thank you. Now, Dr. Suren Pratap Singh, organizer of the event, will introduce to us our honorable guest speaker for today, Dr. Vijay Pratap Singh, sir, Assistant Professor Botany, CMP College, Allahabad. Dr. Suren, please come.
Thank you, Dr. Charu, ma'am. Honorable President ISCA, Professor Vijay Lakshmi, ma'am. Treasurer, Professor Sudhir Kumar Srivastav. The guest speaker, Vijay Pratap Singh, sir, and Dr. Durgesh Kumar, sir. The head of department, Botany, our Jiten Mohan, sir. So thank you, ma'am, for giving me opportunity to introduce our guest, today's guest, Dr. Vijay Pratap Singh. Dr. Vijay Pratap Singh did his BSc in biology from MJP Rohilkhand University, Bareilly. He did his MSc Botany and DPhil with a specialization in plant physiology and biochemistry from the University of Allahabad. Dr. Singh worked as an assistant professor in botany in Government PG College, Chhattisgarh, India from 2012 to 2018. After this, he joined his present institution, that is CMP Degree College, a constituent college of University of Allahabad, as an assistant professor in botany. Dr. Singh has authored 139 original research review and editorials in journals of international repute and 8 book chapters. He has edited about 12 books with academic press elsewhere, Vijay, CRC Press, Springer, Nova Science Publishers, Studium Press, etc. Research projects of various funding agencies like DBT, SERB, and UGC are granted to Dr. Singh, which few of them were completed and few of them are undergoing. Dr. Singh has edited and editing the special issues with the journals such as Journal of Experimental Botany, Frontiers in Plant Sciences, Environmental and Experimental Botany, Physiologia Plantarum, Plant Physiology and Biochemistry, Plant Cell Report, etc. as a guest editor. He is serving as a regular editor of Journal of International Repute such as Acta Physiologia Plantarum of Springer, Plant Growth Regulation, Journal of Plant Growth Regulation, Biologia Plantarum, Biocatalyst and Agriculture Biotechnology of elsewhere, and BMC Plant Biology. His research interests focus on investigation of regulation of physiological, biochemical, and molecular responses in crop plants and cyanobacteria under abiotic stress such as heavy metal, nanoparticles, salinity, ultraviolet B, etc., with an emphasis on reactive oxygen species, nitric oxide, hydrogen sulfide, and phytohormone signaling. Collaborators Analytics has named Dr. Singh among top 1% world most highly cited researcher for the year 2021. Moreover, not only this, the Stanford University has also named Dr. Singh among 2% world's most highly cited researcher from the year 2019, 2020, and 2021. So we have a very big name with us. So today, Dr. Singh will talk on stress adaptation in crop plants. Over to you, sir. You are invited. Just to share the slide.
Thank you, Dr. Suren, and respected Dr. Sudhir Srivastav, sir, Treasurer, Indian Science Congress, Kanpur Chapter, Dr. Jiten Mohan, sir, HOD, Department of Botany, DAV College, my senior colleagues of the DAV College, dear colleagues and dear students. So why we talk about the stage adaptation in crop plants? Because we have 8.5 billion population in 2001, and it will be 8.5 billion by the 2030, and near about 9.7 billion by the 2050. So we have great challenge before us to produce more and more grain for feeding the increasing global population. But in 20th century, due to rapid industrialization, that has resulted in some unwanted changes in our soil and water, that is limiting the overall crop productivity of plants. So we have to seek, or we have to devise certain strategies in order to enhance crop productivity for feeding global increasing population. So my today's talk is based on what type of strategies plants they have adopted to sustain under changing environmental conditions. Generally, a stress that can be defined as any chemical, physical, or biological factor that adversely affects the overall growth performance as well as crop productivity, then we can define it as a stress. And the stress, there are two types of stress: abiotic stress and biotic stress. These are
आज बुलाया है कैसे इसमें पर्सनली बात कर, लेकिन अगर यहाँ की छेड़ी
वाली बात नहीं है, कॉलेज ऑफिस नहीं है, होगी सुविधा सबके लिए होगी, नहीं होगी तो एक के लिए भी नहीं होगी।
यह साथ के लोगों को मना कर दिया
इसका
भाई अपनी नौकरी कौन गवाए,
हमारा क्या दिक्कत है?
कोई बड़ी
यहाँ पे ये
आप एकदम हमारी बात मानिए, विजय जी से आप अकेले जाके बात करते है ना, ना हो समझे किसी लड़के
28 तारीख के लिए
नहीं आज बुलाया है, आज बुलाया है,
बात करें
आज और ना हो तो हम बताएं दूसरे को, है ना। अरे अब आप हम लोग तो पूरी तरह आपके साथ हैं, बुलाते ही क्यों आप? एक काम करो, सुनो है ना, साइन उससे कह देंगे बेटा ये साइन मार देना, है ना।
जी मैडम,
Please unmute yourself.
हाँ बंद कर दिया, बंद कर। वही आवाज चली गई ना, पूछे तो आवाज तो नहीं आ रही है, तो अनम्यूट किया नहीं था हमने।
so
किसने बोल दिया है?
मैडम ने कि आपकी आवाज आ रही थी, हमने बंद कर दिया।
typical definition of stress by distant failure, by stressor, and by larger. I mentioned that there are two categories of stresses. and one is abiotic stage and second one is biotic stage. So these are abiotic states. Temperature may be low and high, water, drought or flood, radiation, whether high like ultraviolet, UV radiation, low or ultra, nutrients, maybe deficiency, maybe excess. Because as far as nutrients they are considered, there are number of nutrients, but only 19 elements so far being selected for basic metabolism, and some element like silicon, selenium, they are emerging as essential elements for plant development, but they are not yet included in the category of essential nutrients. So other elements like arsenic, cadmium, chromium, they are toxic, even they may pose toxicity at very low concentration. As per European Union, only 20 mg per kg of arsenic and 20 mg per kg of chromium is recommended. However, as we are aware that one area is the Kanpur Jajmau, where very high amount of chromium is reported, and in some stretches of West Bengal, where the problem of arsenic is very prominent. So whether deficiency or excess of element, they may cause stress. Biotic, they include animals, pathogens, beads, but my focus of talk is only on abiotic stage. And it is estimated that about 70% of global agriculture land is affected with the biotic as well as abiotic state, that is causing huge loss in crop productivity. So next question is that how these abiotic stresses, they affect overall plant growth and productivity. So there are two indicators, you can say there are two ways through which these abiotic states may induce adverse consequences in plant. One is direct effect, when stress factor like heavy metal, they directly interact with cellular components of the plant and directly affecting the activity of plant. And another aspect is indirect effect. In indirect effect, these abiotic states exerts about toxicity through some other intermediates. So if we talk about the direct effect of abiotic states in plants, so we have taken here as an example of drought state. In the case of drought state, when plant they are exposed to drought state, what kind of responses they occur? First of all, alteration in the gaseous exchange process occurs. Second one is the altered fixation of enzymes related to the photosynthesis, and the negative impact on the constituents of the photosynthesis like photosystem second, and all these effects, they are cumulatively affect the overall response of photosynthesis. And as we know that the photosynthesis is the only process on the earth surface that is responsible for the sustaining of life. And whatever we see, root, leaf, stem, they are all because of effect of or resultant of the photosynthesis. So these are the direct effect of the drought states on the overall crop productivity or plant health. In indirect effect, we talk about the effect of indirect effect of abiotic states. The major mediator of this indirect effect are ROS, reactive oxygen species. And we can say that near about 2.5 billion years ago, when first oxygenic organism, that is cyanobacteria, they were evolved, then this resulted into the induction of free molecular oxygen into the atmosphere. And this induction of atmospheric free molecular oxygen led to the evolution of further organic evolution of plant. Since though oxygen is essential for our survival, but what happens when plants are exposed to abiotic states, as we are all botany faculty are, I think, here, there are two components of photosynthesis, one is light reaction and second one is the a dark reaction. So it's the light reaction which is responsible for the production of ATP and NADPH. So source and sink relation must be maintained. If free NADP plus is not available, then the electron they are released from the photosystem second. Where they will go? They will ultimately received by the molecular oxygen and lead to the formation of superoxide radical. That is one of the negative reactive oxygen species. So these reactive oxygen species like superoxide radical, hydrogen peroxide, hydroxyl radical, they have very high oxidizing potential and they have capability of causing lipid and protein oxidation. So in this way, these are indirect consequences of the stress factors and they are mediated by the reactive oxygen species. In our case of human body, we all know that we are gradually young, then we will be gradually senescent and will old. So in gerontology, that is the science of aging, these reactive oxygen species, they are main culprit for the senescence. So these are the negative aspect of reactive oxygen in the plant system and they are major damaging components when present in the very higher amount. These are the different types of reactive oxygen species like superoxide radical, 1 electron reduction product, then H2O2, 2 electron reduction products, hydroxide radical, they are highly damaging than H2O2 and superoxide radical, 3 electron reduction product, and then the water is formed, that is 4 electron reduction product. These are the reactions which are involved during the induction of formation of reactive oxygen species. So this is the one phase of reactive oxygen species when they are damaging molecule. Another phase of reactive oxygen species is that they act as a signaling molecule. We can't imagine the evolution of plant without the involvement of reactive oxygen species. One example is that we all know that lignin is a very, very crucial component of land plant. And without the evolution of lignin, we can't imagine the evolution of woody plant or land plant. And this lignin is a phenolic compound that is being formed in the phenylalanine pathway, PAL pathway, right? So the polymerization of lignin is occurred in the presence of reactive oxygen species like hydrogen peroxide. So this is a very crucial function of the reactive oxygen species that is performed by the reactive oxygen species like H2O2 is the one example of important aspect of reactive oxygen species. Second one is that hypersensitivity response. As a botany student, we all know hypersensitivity response when an obligate parasite that invades a particular leaf, then the surrounding area of that particular oblique parasite is tends to die through the programmed cell death. And in this fashion, that obligate parasite is restricted in the very small area and remaining leaves structure or surface, they are safe. And in this hypersensitivity response, reactive oxygen species like hydrogen peroxide and one phytohormone that is salicylic acid, that is phenolic hormone, they are major component of hypersensitivity response. And this hypersensitivity response can't be accomplished without the involvement of reactive oxygen species like hydrogen peroxide and salicylic acid. So these two important aspects of salicylic, although we are talking about the negative consequences of reactive oxygen species in plant, but we have cited these two signaling molecule behavior of reactive oxygen species to remember that all reactive oxygen species, all time, they are not harmful. And their concentration in the plant system or animal system is controlled by the antioxidant defense system. Their spatial and temporal generation is required for the overall growth performance of the plant. So on what level these abiotic stages they affect plant? One, at growth level, they affect seed germination, they cause reduction in growth, they cause premature senescence. And at physiological level, they alter with the water uptake pattern, they alter transpiration rate, they alter respiration, they alter nitrogen assimilation. As we know that there are 5 enzymes in nitrogen assimilation like nitrate reductase, nitrite reductase, glutamine synthetase, glutamine 2 oxoglutarate, aminotransferase. All these enzymes, they are somehow affected under abiotic state. And third one is molecular level. How abiotic state they affect plant at molecular level, they alter gene expression. For example, they alter the expression of enzymes. They are encoding for Calvin cycle enzyme. Photosynthesis will be declined. They start breakdown of macromolecule. They reduce the activity of vital enzymes like Krebs cycle and photosynthesis Calvin cycle. They may decrease protein synthesis, and ultimately they may disorganize membrane system. Respected seniors and dear colleagues, we must remember that our entire system, whether plants or animals, they are somehow depend upon the endomembrane system, like plasma membrane and cellular organelles. So integrity of the membrane system is very essential for the survival. So now question is that how to address with these abiotic states and what stage they are involved in the stage sensing and adaptation. First, during adaptation and acclimation, though parallel terms, but they are somehow different. In adaptation, while changes, they are genetically fixed. However, in acclimatization, they are somehow permanently not genetically fixed, but for same generation, they have somehow registered abiotic stage. So first step is that the stage is sensed by the plant system. For example, we have taken example of sodium chloride stage, NaCl stage. First, these sodium and chloride ion, they are sensed by the plant. Oh, if they are in sodium or chloride ion, they are first sensed by the plasma membrane. Then downstream signaling cascade, they are started. What happened? When sodium chloride stage is exposed to plant, potassium by sodium ratio is very high. That is not better for the plant. So what strategy plant they adopt? They adopt to increase the pumping of more potassium ion from the soil to the plant system. In this way, they maintain their osmotic balance. So sensing is essential, then followed by downstream signaling cascade like activation of plasma membrane bound potassium channel, and these two cumulative responses, they result into the adaptation. Finally, plant they adapt themselves against the sodium chloride stage. So there are two strategies where plant adapt to these abiotic stage. One is the inherent strategy. The inherent adaptation strategy of plant. They are inbuilt within the plant system. And second, by using some other means like biostimulant, fertilizer, or some other phytohormone or signaling hormone. So these two, through these two components, we can produce more adaptable plants and with higher productivity. So in first category, inherent adaptation, how plant they have inbuilt adaptation strategy. So this is the one example of inherent adaptation strategy at physiological level. As we know that photosynthesis completes in two steps. First, photochemical reaction, and second one is the Calvin cycle. So in chemical reaction, photosystems are involved, photosystem second and photosystem first. There are three pigments and different electron carriers. In photosystem second, this is protein pigment complex, and this is made up of proteins and pigments. And out of these proteins, D1 protein is very necessary. D1 protein is very necessary as well as level to the stress condition. As we know that plants, they are sessile. They can't move from one place to another place. Suppose in May and June month, when light intensity is very high, what plant will do? They can't move from one place to another place. So in this situation, while light intensity is very high, that is continuously impinging on the photosystems. So in order to channelize this light energy, D1 protein is somehow damaged or degraded. But one of the peculiarity of the system is that this damaged D1 protein is automatically synthesized, or you can say de novo synthesized. So in this fashion, plant they protect themselves from the high light intensity by the D1 protein turnover. The plant can be withstand high light states. This is the second example. Koji Athada and no more who proposed the cycle, water cycle. As we know that oxygen evolving is complex associated with photosystem second, light intensity is very high. Electrons, they are coming out from the water and ultimately reach to the NADP plus, resulting into the formation of NADP plus, NADPH. When light intensity high, perhaps some enzymes of Calvin cycle, they get inactivated. NADP plus is not recycled. Then where electron will go? Then electron are received by the molecular oxygen that has high electronegativity and resulting into the formation of superoxide radical. Superoxide radical, they are damaging and simultaneously disproportionated to the H2O2, less toxic H2O2 by the enzyme superoxide dismutase. H2O2 is still toxic and it is dissociated into water and oxygen with the help of enzyme APX, ascorbate peroxidase or catalase. So enzymes, they are electrons, they are coming out from the water, they are again incorporated into water. That is why we call it as a water cycle, and it is a very, very crucial cycle for dissipation of excess photons, or we can say for the mitigation of high light intensity, particularly during the May and June. In this way, plants they protect from the high light intensity through these two processes, D1 protein turnover and water cycle. And this is the second adaptive strategy of plant at chemical level. Here we have taken only one example of ascorbate glutathione cycle. The discoverer ascorbate Asada Halival cycle. This cycle is also called as Asada Halival cycle. This cycle comprises of 4 enzymes: APX, MDHAR, DHAR, and GR, and 2 metabolites, ascorbate and glutathione. Respected seniors, we have to say that this ascorbate and glutathione, they are known as a redox buffer of cell. We can't imagine of sustaining of our life without these two molecules, and their concentration in the cellular system exists in millimolar concentration. So they are actually redox buffer of the cell. So these are 2 non-enzymatic part and 4 enzymatic part. They comprise of this ascorbate glutathione cycle. This cycle is start with the scavenging of H2O2 with the help of APX enzyme using ascorbate as an electron donor. And then this cycle is started and passes through MDHAR, DHAR, and culminates with the regeneration of reduced glutathione. So there are 6 components, and these 6 components we can utilize to generate more efficient plant. They can produce more grains, more pulse under changing environmental conditions. I think Surendra can optimize these 6 components for producing more climate resilient plants. And third one is at the molecular level. At molecular level, what strategies plant adapt to sustain under changing environmental conditions? We have taken as an example of drought. Under drought condition, what happens? Under drought condition, drought is sensed by the plant cell, and it is accompanied by the more and more production of abscisic acid. Abscisic acid is perceived by the receptor paravectin-like resistance 1 protein, and this protein leads to the activation of a small nuclear receptor kinase. And this receptor kinase activates pumping of more potassium ions from guard cell to the subsidiary cell, resulting into the closure of stomata. By closing the stomata, plants they can survive under a drought state condition. And another important example of drought state is that if we are going to grow one plant under well water condition, another plant under drought state condition, then we will observe that roots will be longer under drought state condition than the well water condition. So this may be another strategy. And second one is that if we are going to sow seeds in drought state condition, then seed will not germinate. because there is no future of seed to grow under drought state, and it is sensed by the seed itself. However, if moisture is there, no problem. Acetic acid will be degraded and gibberellic acid will be produced and germination will occur. And last, how we can enhance efficiency of plant under environmental stress condition in order to produce more grains and other food commodity. So we have taken here one example of silicon fertilizer. Although silicon is not necessary for the plant, but we have to say that the maximum members of POC, they can even not complete their life cycle without silicon, because silicon is necessary component of their life cycle. And another aspect that we know that carbon life is exist on this earth surface, but it is also not a wonder that silicon life may exist some harass condition somewhere else. Silicon and carbon, although they are sister elements, but silicon is the more rigid than the carbon. That is why carbon is being selected for the organic evolution, but silicon is not. But silicon life may also exist like other harsh conditions like Mars and some other in low basin of the ocean. So we have taken silicon as an example. How can we enhance crop productivity under metal stress condition? One metal we have grown plant under metal stress, primary roots, they are reduced in size. If we are going to expose plant to the heavy metal stress, the length of primary roots may decline, but root hairs may increase. That may be adaptive strategy, because if we are going to give 1 mg poison to 10% and 1 mg poison to the 100%, then what will happen? 100% will tolerate efficiently that 1 mg poison, but I think 10% will not. So by increasing the root hair formation, plant may sustain their life. So silicon promote root hair formation under metal stress. I have also studied and published some good paper in the international journal. So in this way, silicon, very cheap component. We may also propose silicon as a fertilizer in the agricultural field. Sulfur, we have studied effect of sulfur. A Jajmau area is very polluted with chromium. Although transporters, they are not being evolved for the transport of non-essential elements, but some structural similarity they show with other metals like chromium shows a structural similarity with the sulfur transporters. Arsenate shows a structural similarity with phosphorus. Cadmium shows a structural similarity with the zinc. So we can increase crop productivity by increasing the amount of silicon and sulfur in surrounding medium of the crop. And in this way, we can protect plant health as well as increase crop productivity in the agro ecosystem. So what happens when we provide silicon under heavy metal stress? It regulates water uptake, induces more and more root formation, balances mineral uptake, adjusts osmotic adjustment, increase activity of enzymes and photochemical efficiency, and improves antioxidant defense system. So there are 3 inherent capacity, and why this externally added component we can increase crop productivity, like phytohormones like carnitine, indolestic acid, they are very cheap. We can utilize these components. Silicon is very cheap, sulfur is very cheap. Even donors of signaling molecule, nitric oxide, hydrogen sulfide, they are very cheap. So we can use these components in our agro ecosystem. Thank you. And these are 4 PhD students. They are working under my supervision. Here is Vipul Misra. He has published 2 papers in Trends in Plant Science, having 22 impact factor. And these are 3 new students. They have enrolled in this session under my supervision. Thank you very much. आप लोगों से कोई क्वेश्चन हो तो बताइए. कितना मिनट का 40 मिनट वहाँ हुआ। कितने मिनट का है? हो गया, ठीक है।
Any questions? Please unmute yourself or write in a chat box. No questions, so proceed to next.
हाँ, सिलिकॉन, आई थिंक सिलिकॉन का रोल ज़्यादा नहीं है पोटैटो में। पोएसी के बहुत से मेंबर्स हैं, राइस है, व्हीट है, मेज है, शुगर केन है, उनमें बहुत ज़्यादा सिलिकॉन। अगर समहाउ देखिए दो रिस्पांस होते हैं, अगर आप हाई डोज में यूज करेंगे देन विल काज इनहिबिशन, एंड एट लो डोज देन इट विल काज ग्रोथ इंड्यूशन। सो इट इज हार्मोनिक इफेक्ट। आप यूज कर सकते हैं, कम अमाउंट में यूज कर सकते हैं, डेफिनेटली वो ग्रोथ इनहांस करेगा। लो प्लांट के लो टेम्परेचर के लिए ऐसा तो नहीं है, लेकिन अगर हम उसी के अकॉर्डिंग कुछ रेजिलिएंट कुछ कंपोनेंट अगर उसमें हम सर्च करें, जैसे मैंने आपको जो बताया है, दे आर फंक्शनल बायोमार्कर, जैसे मैं 6 बता रहा था ना आपको, दे आर फंक्शनल मार्कर, वी कैन इंजीनियर देम या वी कैन समहाउ एडजस्ट देम अकॉर्डिंग टू द नीड ऑफ क्रॉप प्लांट। तो ऐसा कुछ अगर उसमें बायोमार्कर होंगे तो जरूर कुछ ना कुछ हम कर सकते हैं। और वैसे भी आलू हमारे यहाँ का नहीं है, तो हम लोग उसे ठंड में यहाँ ग्रो कराते हैं। देखिए अब ये डिपेंड करता है, प्लांट की इफिशियंसी पे भी डिपेंड करता है कि कौन प्लांट किस इफिशियंसी के साथ टेकअप करता है। जैसे फॉर एग्जांपल मैं आपको बता रहा हूँ, अभी नैनो फर्टिलाइजर में एक नैनो यूरिया आई है, नैनो यूरिया अभी इफ्को ने रिलीज की है। तो वो ये कहते हैं कि हमारी 500 ml नैनो यूरिया, जो 50 kg का पैक आता था ना पहले यूरिया का, उनकी 500 ml नैनो यूरिया 1000 ppm इज इफेक्टिव, एज मच इफेक्टिव दैन 50 kg। तो कुछ कर सकते हैं हम, नैनो ला सकते हैं उसको, लेकिन ये डिपेंड करता है हमारे प्लांट की इफिशियंसी पे, एंड इट वेरीज स्पीसीज टू स्पीसीज, इवन जेनरा टू जेनरा। हाँ,
थैंक यू सो मच सर। आपने बहुत ही बृहद ज्ञान हमारे स्टूडेंट्स को दिया और हम लोग इस संदेश से लाभान्वित होंगे। Now our next esteemed invited speaker is Dr. Durgesh Tripathi sir from Amity University, New Delhi. Dr. Surendra Pratap Singh will give some introduction about Dr. Tripathi. Over to you, Surendra.
Thank you, Dr. Charu. We have with us Dr. Durgesh sir. Okay, so sir is online.
Yes, Dr. Suren, thank you so much. I can hear you, and yes, I'm actually I'm in your lectures from the very beginning. Thank you so much. Please start.
So Dr. Charu has given me the word to introduce Dr. Durgesh sir. Dr. Durgesh Kumar Tripathi is a senior assistant professor and joint coordinator research and development at Amity Institute of Organic Agriculture, Amity University, Noida. Dr. Tripathi did his MSc from DDU Gorakhpur University, Gorakhpur, and has received his DPhil in Science from University of Allahabad, Prayagraj. He is the recipient of DS Kothari Postdoctoral Fellowship of UGC New Delhi and has completed his postdoctoral fellowship tenure from the Center of Advanced Study in Botany, Banaras Hindu University, Varanasi, India. Dr. Tripathi has worked extensively on heavy metal and toxic nanoparticles detoxification and has elaborated tolerance mechanism in plants like rice, wheat, maize, tomato, brassica, which could help in improving crop productivity, benefiting for the society. In nutshell, his research interest lies in a crop nanobiology, crop stress physiology, nitric oxide, reactive oxygen species, hydrogen sulfide signaling, phytohormone crosstalk, plant anatomy, and root biology, nutrient and nanonutrient regulation in plant life. He has published more than 175 research and review papers in the Journal of International Repute and has edited 16 books with a renowned international publisher like Elsewhere, Springer, Vijay, CRC Press. Dr. Tripathi is the recipient of several national and international awards, including Tony B. Academic Travel Award, Washington, DC, US, top 1% research around globe by Collaborators. This is a big achievement for us. Web of Science, USA, top 2% researcher around the globe by Stanford University, another big for, and a Young Scientist Award in 2019. He is the life member of various national and international scientific societies like Indian Science Congress, Association India, International Society for Silicon in Agriculture and Related Disciplines, and International Society of Root Research, etc. So today, Dr. Tripathi will enlighten us on a topic, small or big, nano potential to enhance the plant resistance against stress. So, sir, over to you. I'm making you the host. You can share your screen.
Okay. Thank you so much, Dr. Suren, for your for my nice introduction. Thank you so much. I just sharing my slides, but I'm not able to slide.
I'm making you host. Okay. Okay, sir. Now you can share.
Okay. Is it visible, Dr. Suren?
Yes, it is visible, sir.
Okay, I'm sorry. Actually, I have slightly changed my my topic. I don't. I think it is not a big issue. I just slightly changed my topic.
Okay, sir. Over to you, sir. Please.
Okay. Okay. So thank you so much, Dr. Surendra, and I truly feel honored and privileged that to get the opportunity to be a part of this prestigious Dr. Nagin Swarup Memorial Lecture Series 2022. I want to express my heartful gratitude to Professor Arun Kumar Dixit sir, Principal DAV College Kanpur, Dr. Vijay Lakshmi Saxena, ma'am, President Indian Science Congress Association, Dr. Sudhir Kumar Srivastav, Treasurer ISKA Kanpur chapter, and Dean Faculty of, and also the Dean Faculty of Science and CSGM, and local secretaries of Indian Science Congress, Dr. Suren Kumar Singh sir, and event organizer, and Dr. Vijay Pratap Singh and other dignitaries. Thank you so much for providing me this wonderful opportunity to speak in this Science National Science Congress local chapter. The topic of my lecture is nano potential in agriculture and stress resistance to the very small. Nanotechnology is a very nowadays very exciting research topic. There are all over the world people are focusing so much to utilize these small particles to take more benefits for the agriculture, for the medical science, and so many other allied sciences. So because my lecture is basically based on the agriculture and stress resistance, so first we'd like to start from two major questions: why we need to improve the agriculture. This is the first major question: why we are focusing so much on the agriculture improvement. And second is how nanotechnology can address this issue, right? Because we have certain so many things to utilize, like biotechnology, molecular biology, to improve our agriculture. But why we are focusing so much on the nanotechnology? So these are two major questions, and so we will discuss basically on these two major questions. Is my lecture is visible, Dr. Suren?
Yes,
My
sir.
slides are visible?
Yes, sir, it is visible.
Okay, thank you. So we all know that the present global population of 7.7 billion is projected to grow nearly sorry, there is some issues in my PPT, right? So yes, we are going to discuss why we need to improve the agriculture, how nanotechnology can address these issues. So we all know that our global population is increasing day by day, and we need a sufficient amount of food till 2020, till 2050, and to resolve this issue, scientists of all over the world and governments of all over the countries, they are focusing so much to improve the agriculture, because we all know that the agriculture land is reducing day by day due to the industrialization, right? So to resolve this issue, the expert of different fields, they are like some people, they are working on abiotic stress, some people are working on microbiology, some people are working in molecular biology, and utilizing a very advanced technology to improve the agriculture. So through this way, in a very similar fashion, nanotechnologies, they are also working to produce some kind of nano fertilizer, some kind of nano solution which could be helpful to improve the agriculture, right? So nanotechnology could make agriculture more efficient and more sustainable, but more systematic understanding of the mechanism involved is necessary to prove the potential of nano enabled product. So before starting this, we should know about what are the nanoparticles or nanomaterials. So nanomaterials are generally classified as those material having at least the one dimension less than 100 nanometer, which leads to the possibility that they can exhibit different properties than the respective bulk material. You can see here the different kind of and different signs of different size of the nano materials. Engineered nanomaterials are materials created with the nanoscale dimension. They are over 1,800 consumer products reported to, including environmental nanomaterials. For example, you can see here in this picture application of nanoparticles in different fields, right? Like medical science, engineering, agriculture science, and so many other disciplines. There are many applications they are possible due to the application of nanoparticles. Also, there have been a great advance in the production and use of Indian nanoparticles. Their impact on health and the environment are not completely clear, and often depends on the transport and exposure pathway, as well as the subject into which in Indian nanoparticle enters. The complexity regarding behavior of nanoparticle often leads to seemingly conflicting conclusion, causing confusions among scientists and especially among the non-scientists, because there are many reports which clearly suggested that nanoparticles somewhere are very toxic, and some studies, they are suggesting that nanoparticles, they are very beneficial for the plant growth development and abiotic stress management. So there are so many views till date about the impact of nanoparticles. So first we need to know about how they enter into the plant system, there are different ways, like our ion transport is possible. So nanoparticle also take the same routes, like apoplastic pathway, symplastic pathway, and so through these way these small nanoparticles, they enter into the plant cell, and then they start reacting with the plants, like several tissues, like epidermis, endodermis, and barrier Casparian stem must be crossed before the reaching the vascular tissues, depend on the entry point. A small size of the nanoparticles allow their entry through cell wall pores. And the next is internalization of nanoparticle inside the cell, which proposed to occur through various mechanisms, uh, endocytes, pore formation mediated by the carrier protein, and through plasmodesmata. Nanomaterials follow the apoplastic or the symplastic pathway for moving up and down the plant, and radial movement for the changing from one pathway to the others. With all these nanoparticles being produced and entering the environment, it seems reasonable for us how these nanoparticles will affect the environment and different life forms, including the plants. So this is the major question, and we should know about how nanoparticles they behave with the different living systems. So we have now three components: nanoparticle toxicity, because there are thousands of studies clearly demonstrated that nanoparticle, they are very much toxic. Then again a question arises that if they are toxic, then how they are helpful for the plant growth development and agriculture in an agriculture perspective. So we will also also discuss a second component: nanoparticle induced toxicity alleviation in plant. If nanoparticle they are toxic, then how we will manage the nanoparticle toxicity. The third component is nanoparticle can tackle the stress in plants, because as on the basis of different studies that nanoparticle, they are very much beneficial and they can play a greater role to control the abiotic stress. So we'll also discuss these things in our component third. Now the component first, we will start nanoparticle toxicity, because the different studies they have shown that nanoparticle they are very much toxic. So we have initiated a study which is published in Ecotoxicology and Environmental Safety in 2019, that liquid assisted pulse laser ablation synthesized copper oxide nanoparticle and their different impact on the rice seedling. So we have utilized a green technology to synthesize the nanomaterial, like LIBS. We have utilized the LIBS, laser induced breakdown spectroscopy, and we have synthesized the copper nanoparticle through this very advanced technology. This research involved the synthesis of laser ablated copper nanoparticle in liquid medium, analyze their properties, accumulation behavior, and impact on the physiological and biochemical parameters of the rice seedling. Then what are the key results we have found? So we have found that lower concentration, means 5, 10, 20, and 50 micromole, the copper nanoparticle enhance the growth in terms of fresh weight, dry weight, and length, photosynthesis, pigment, and other biochemical parameters, including some photosynthetic parameter like fluorescence parameter, and the amount of some mineral nutrient like calcium, magnesium, sodium, and potassium in rice seedling. But it happens only at the lower concentration of the copper nanoparticle. However, higher concentration like 100, 200, and 500 micromole copper nanoparticle cause decrease in these parameters, means ki growth and chlorophyll, carotenoid, and mineral nutrients, right, decrease these parameters due to the enhanced level of copper nanoparticle. Because at lower concentration, we have found that the accumulation of copper nanoparticle is increased. The increased rate of copper nanoparticle is very high, which which result the enhanced level of oxidative stress marker such as hydrogen peroxide and MDA. These research suggests that eco-friendly laser ablation synthesis method is comparatively easy to many advantages over other chemical route, that is formation of controlled size nanoparticle with controlled properties without any contamination. The data clearly demonstrated that lower concentration of copper nanoparticles show positive effect and improve the growth of rice cereal. these are the some other results. This is the, you can see here the laser induced breakdown spectroscopy setup, and these are some liquid medium in which we have synthesized the nanoparticle. This is the physiological and biochemical results which we have given in our paper, which is already published in Ecotoxicology and Environmental Safety. So the study present that lower concentration of pulse laser ablation copper nanoparticle might be beneficial for growth and development of rice seedling. Higher concentration of copper nanoparticles show toxic effect and thus hamper growth by producing oxidative stress as a result of higher uptake of copper. This work opened possibility for more extensive research on laser ablated synthesized nanoparticle and their interaction with the plant. Basically, scientists utilize the chemical synthesis of the nanoparticle, or they utilize the plant extract, and then they synthesize the nanoparticle. But laser induced synthesis of nanoparticle is one of another and very interesting technique which may be helpful to control the contamination of the nanoparticle synthesis. This is another study which is of my lab, which is published in Photosynthetica. This study presents a mechanism of silver nanoparticle action on the photosynthetic process and induction of the oxidative damage in chloroplast of the plant cell. The two concentration of, in this study we have taken two concentration, 1 and 5 micromole, showed negative impact on the growth parameter such as root and shoot length and weed seedling. In addition, the biochemical parameters such as activity of catalase.
Sorry to interrupt you. Can the PPT go full size mode?
Yes, yes, yes.
Yeah, this will be visible, I think. Yeah.
Okay.
Oh.
Now it is visible.
Okay. Yeah, it's look good. Thank you, sir.
Okay. So yeah. So yeah. In addition, the biochemical parameters such as activity of catalase, glutathione, flavonoid, and chloroplast concentration were also affected. We have measured in our lab. This was a collaborative study. So there are some scientists from the Slovakia and Poland were associated with my group. So we have found these results. So AGNP, next is AGNP apparently suppress the photosynthetic activity of seedling with a clear distribution of photosystem at 5 micromole concentration. So these are some results which we have observed during our entire studies. Here you can see. And what are the conclusion of this study? This research indicates that photosynthetic phytotoxic effect of silver nanoparticle on growth of yeast seedling, and this involved an improper regulation of PS first electron transport, resulting in a damage of chloroplast structure, including photosystem itself. It showed that the photochemistry, especially the process at the level of photosystem first, is severely affected by the AGNP treatment. The acceptor site of photosystem first can be major source of free radicals, which are responsible for oxidative damage observed in this study. Now, so in first component, we have discussed about the toxicity of the nanoparticle, which we have a study in our lab or with along with our collaborators. Now the second component is that how we can control the nanoparticle induced toxicity in the plants. So now we will discuss in this component that how we can if we find the toxicity of nanoparticle, because we all know that nanoparticle, they are more efficacious than their bulk material. So if the nanoparticle concentration will enhance in near future in our agriculture field, so it will be more, so these nanoparticles will be more dangerous for our soil, for our crops, as compared to their bulk material. So we have some kind of solution that if we find some toxicity of any nanoparticle in our agriculture field, then how we can control these nanoparticle induced toxicity in our agriculture field. So now in this section we will discuss that what are the major components through which we can control the nanoparticle toxicity. So here this study we have published in Plant Physiology Biochemistry, in which we have studied that nitric oxide can alleviate silver nanoparticle induced phytotoxicity in Poison sativum seedling. This study was conducted to investigate the adverse effect of silver nanoparticle on pea seedling, and nitric oxide being an important signaling molecule and capable of enhancing plant tolerance to different environmental stress, was assessed for its potential to mitigate toxicity of AGNP. So what are our key findings in this study? That AGNP treatment 1000 at 1000 3000 micromole significantly declined the growth parameters and the photosynthetic parameter and some biochemical parameters, right? So, but addition of SNP means ki sodium nitroprusside, which is the donor of nitric oxide, mediated AGNP-induced adverse effect on these parameters, at it's reduced the accumulation of silver and repaired the anatomical structures. So these are some results which we have discussed in our study, and we have found here, as you can see, that how AGNP significantly decrease the overall length, fresh mass and dry mass. However, addition of nitric oxide again manage these toxicity and protect the plants against the silver nanoparticle stress. These are some results which we have discussed in our study. These are some anatomical parameters. We have cut the section of root and shoot and observe that what kind of changes the plant can analyze. We can find what kind of changes due to the exposure of AGNP in the plant, root and shoot, and how these adverse due to the adverse effect, how nitric oxide can manage these adverse effects. What are the conclusion of this study? That this study confirmed the phytotoxicity of silver nanoparticle treatment and 1000 and 3000 micromole pea seedling. It also validated the capability of nitric oxide, an important signaling molecule which mitigate silver nanoparticle induced toxicity. Mitigate AGNP induced toxicity, as SNP addition was found to regulate the silver uptake, antioxidant system, oxidative stress, and anatomical structure of the root and shoot. So through this way, application of nitric oxide, we can protect the plant from the silver nanoparticle toxicity. This is another study which is published in very recently in Journal of Hazardous Material. Hydrogen sulfide and other signaling molecule, which is very beneficial to control the copper oxide nanoparticle induced phytotoxicity in rice or Rhiza sativa seedlings, and also support silicon, because as Dr. Vijay has discussed in his lecture, that silicon is a very beneficial element for the plant growth and development, and it also very effective against the abiotic stress. So we have also analyzed the what is the mechanism work behind when we utilize the silicon against any abiotic stress. What mechanism work behind this abiotic stress tolerance because of application of silicon? We have found that hydrogen sulfide helps helps silicon to mitigate the any kind of abiotic stress. But in this study, we have only focused that how hydrogen sulfide underpins the beneficial silicon effect against the copper oxide nanoparticle. So this study evaluated whether the exogenous hydrogen sulfide application in combination with silicon could mitigate the harmful effect of copper oxide nanoparticle in rice seedling. So this is overall our experimental setup, which we have set up in our lab, and we have analyzed the plant. These are the rice plant under different exposure of silicon, hydrogen sulfide, and copper oxide nanoparticle, and we have analyzed the data. So what are our key results? Exogenous application, we have found that exogenous supplementation of 10 micromole silicon and 100 micromole NaSH, means hydrogen sulfide, which is the H2S donor, simultaneously provided tolerance and enhanced defense mechanism of the rice seedling against the 100 micromole copper nanoparticle. It was observed in terms of their growth, photosynthetic pigment, antioxidant enzyme activities, the content of non-enzymatic components, chlorophyll, fluorescence, and other biochemical parameters, and we have found a very exciting results. Also, silicon and NaSH stimulate gene expression of silicon, LSI1 and LSI2 transporter gene, which are the transporter gene of the silicon, LSI1 and LSI2. and auxin, pin 5 and pin 10 transporter genes. So we have also analyzed these the expression level of these genes. These results suggest that hydrogen sulfide might be involved in SI-mediated mitigation of copper nanoparticle stress in rice seedling. These are some our we have analyzed some biochemical parameter, oxidative stress markers, and overall nitric oxide production, histochemical analysis. These are some antioxidant enzymes results which we have discussed in our studies, and this is our overall mechanism. So what are the conclusion of this study is this research identified hydrogen sulfide to assist silicon in order to eliminate copper oxide nanoparticle toxicity in rice seedling. It means silicon take help from the hydrogen sulfide to control the toxicity of copper nanoparticle. S2S underpin the beneficial silicon effect against the CuNP induced oxidative stress and stimulation of the enzymatic component of ascorbate glutathione cycle in rice seedling. Similarly, this is another study in which we have analyzed silicon and plant growth promoting rhizobacteria differentially regulate silver nanoparticle induced toxicity in Brassica gentiana application of nitric oxide. This study we have analyzed along with Dr. Vijay Pratap Singh, which is which is your another guest, and in the collaboration of Professor C.V. Sharma from MNIT Allahabad. And this study was carried out to investigate the role of silicon and PGPR and phytohormone indole acid in regulating AGNP stress in Brassica gentiana seedling. These are our major findings in which we have analyzed that how PGPR and silicon nanoparticle they are very much helpful to control the overall toxicity of the silver nanoparticle. These are some histochemical analysis which analyze that how when we provide the silver nanoparticle, what are the expression level of these oxidative stress marker. You will see that how these oxidative stress markers they are increased very much under the application of silver nanoparticle. However, when we provide the silicon alone and as well as the silicon along with the PGPRs, we found a very interesting result, and silver nanoparticle along with the PGPRs very much helpful to control the silver nanoparticle toxicity. So silver nanoparticle showed significant reduction in growth parameters due to the greater accumulation of silver ions in seedling that lead to reduce the capacity of photosynthetic efficacy, oxidative stress, and other biochemical parameters. These effects were accompanied by the increasing accumulation of nitric oxide. However, interestingly, silicon when present with AGNP enhance its toxicity, means silicon alone is not capable to control the toxicity of silver nanoparticle, and it also reduce the growth and mechanistic properties of Brassica gentiana together with high level of NO. Thus, NO act as a positive regulator of silver nanoparticle toxicity. However, PGPR addition with the AGNP cause significant alleviation in detrimental effect of AGNP on Brassica gentiana seedling along with the reduced level of NO. This is overall mechanism which we have proposed in our study and which is published in very high impact factor journal in Journal of Hazardous Material. So this is the conclusion of our study, that this study suggests the formulation and utilization of combination of silicon and plant growth promoting rhizobacteria as biofertilizer for eradicating the stress in plant. This conclusion was based on the reversal of the silver nanoparticle toxicity in Brassica gentiana seedling by combined application of PGPR and silicon. This is another study which we have applied the endogenous indole acetic acid and nitric oxide are required for the calcium mediated elimination of copper oxide nanoparticle toxicity in wheat seedling. We have found in an effort of the minimizing the copper nanoparticle toxicity, exogenously applied calcium was assessed for the potential to the toxicity of copper nanoparticle in wheat seedling. We have found that calcium is very much effective to control the copper oxide nanoparticle toxicity. These are some biochemical results, oxidative stress markers we have analyzed, and in which we have clearly see that how calcium is very much effective to control the toxicity of copper nanoparticle. This is overall mechanism which we have proposed in this publication. This is published in Physiology of Plant in 2021. This research delineated the mechanism through which calcium mediated copper nanoparticle toxicity tolerance in wheat seedling. The addition of calcium protects wheat seedling against copper oxide nanoparticle toxicity by maintaining endogenous NO, and in low lactic acid maintaining the redox status of ascorbate and glutathione and lowering the accumulation of copper and oxidative stress marker. Now the most important component third. So I will not take so much of time. I will just compile our overall lecture in this component, which is very important for all of us and for those scientists, for those farmers. They are struggling to control the abiotic stress. They are struggling for a greater solution and so that they can improve the overall production of the agriculture. So this is the component third, in which we discuss nanoparticle can tackle the abiotic stress in plant. So there are so many nanoparticles which are very beneficial, which have capacity to protect the plant. So a new route for plant nutrient delivery against the abiotic stress. So here we can see that we have applied silicon nanoparticle alleviate chromium phytotoxicity in Pisum sativum seedling. This research was conducted to confirm the beneficial role of silicon against the heavy metal stress, right? So we have just synthesized the silicon nanoparticle. We have converted the silicon into the silicon nanoparticle through the very green synthesis. The study involved analysis of growth parameter, photosynthetic and biochemical parameters against the chromium toxicity in pea seedling. And what are the key results? That we have found that, as it is well known that chromium is very much toxic to the plants. So chromium reduce the growth, and because of its higher accumulation in plant cells, decrease the chlorophyll fluorescence, increase the superoxide radicals, increase the antioxidant activity of superoxide dismutase and ascorbate peroxidase, and decrease the micro and macronutrient content. However, addition of silicon nanoparticle along with the chromium, it decrease the chromium accumulation, increase the photosynthetic pigments, and increase chlorophyll fluorescence and other very important biochemical stress markers, as well as increase micro and macronutrients. And through this way, silicon nanoparticle, we have found they are much capable to control the chromium toxicity after the accumulation of this after the application of against the chromium stress. Here these are the results related to fresh mass, dry mass, length, and these are the antioxidant results, in which we have clearly seen that what are the changes after the application of the chromium we have found, and how the silicon is much capable to control the the toxicity of the chromium. So this study suggests that 4 mechanisms by which silicon nanoparticle alleviated chromium nanoparticle induced toxicity. First, reduction of accumulation of chromium in root and shoot. Second is due to the decreased accumulation of chromium, a reduction in level of ROS, upregulation of antioxidant defense system, which effectively regulate oxidative stress, and upregulation of uptake of mineral nutrient, which provides protection against the oxidative stress. So this study may provide a basis for the understanding the role of silicon nanoparticle in alleviation of metal toxicity, and suggested about application of silicon nanoparticle against the risk associated with heavy metal toxicity and sustainable plant production. Next study we have similarly we have analyzed silicon nanoparticle more effectively alleviate the arsenic toxicity than silicon in maize cultivar, and hybrid different in arsenic tolerance. So in this study we have taken 2 types of the maize. First one is cultivar and second one is a hybrid, and we have applied silicon as a bulk material as well as a silicon nanoparticle to compare whether which form of the silicon is more effective against the arsenic toxicity in two type of the maize cultivar. So this is as compared silicon nanoparticle and for their potential to and silicon for their potential against the mitigate arsenic toxicity in maize cultivar and hybrid differing the air arsenic tolerance. So these are our finding that we have applied 25 and 50 micromole concentration of arsenic and 10 micromole silicon and 10 micromole silicon nanoparticle to see what are the effect of the silicon and silicon nanoparticle against the arsenic toxicity, and through this we have analyzed different parameter that we have see that what are the concentration of the arsenic concentrate, or what are the concentration what are the content of arsenic when we give application, when we provide the application of arsenic alone, as well as combination in silicon and silicon nanoparticles. These are some oxidative stress marker. What are the conclusion of this study? That's comparing result of silicon and silicon nanoparticle is what observed that reduction in the accumulation of arsenic and the oxidative stress markers were higher under the silicon nanoparticle and arsenic combination than silicon, which is due to the greater availability of silicon nanoparticle in seedling than silicon, because silicon nanoparticle they are much capable to induce the cycle, where higher in silicon in a silicon nanoparticle and arsenic combination than silicon, which subsequently counterbalance ROS mediated damage to macromolecules more efficiently. Therefore, this study established that silicon nanoparticles are more effective in reducing arsenic toxicity than the silicon. So similarly, this is another study which we have conducted on silicon nanoparticle more effectively alleviate UV stress than silicon in wheat seedling, and we have found a similar kind of result that because we that ultraviolet B radiation is another abiotic stress. So we have just want to confirm that whether it is only possible in silicon nanoparticle, they are much capable only against the heavy metal stress or any other abiotic stress. So we have found that silicon nanoparticle not only effective against the heavy metal toxicity, as well as it is also very much effective against the ultraviolet B radiation. And in this study, we have also found a similar kind of result that that silicon nanoparticle is more effective to control the ultraviolet B radiation against the to control the ultraviolet B radiation as compared to the silicon, which is the bulk material. So these are some results, and we can see that how these results, first mass, dye mass, these are some growth related parameters. We can see that how silicon nanoparticle more effectively protect the plants against the UVB radiation as compared to the silicon alone. So what is the conclusion of this study? Conclusion is the same, that silicon nanoparticle, instead of utilizing the silicon as a bulk material, we should utilize the silicon nanoparticle so that we could be more effectively control any abiotic stress of the plant system. This is the another and the last study of this presentation, of my presentation, in which we have utilized the silicon nanoparticle and indole acetic acid in alleviation of the chromium toxicity in Oryza sativa seedling. We have utilized, we have just combined with indole acetic acid to find out what mechanism work behind the silicon nanoparticle application, and how the silicon nanoparticle, they are more effective to control any kind of abiotic stress, whether other signaling molecule or phytohormones, they are also involved, they are also support this in this overall this overall control. So this study investigated eliminative effect of silicon nanoparticle and indole acetic acid alone, and in combination against hexaviolet chromium toxicity. We have found that 100 micromole chromium caused toxic effect in rice seedling at morphology biochemical parameter. However, SINP and indole acetic acid application to plant under chromium toxicity stress enhance their tolerance and defense mechanism as manifested in the terms of increased biomass and endogenous NO, photosynthetic pigments, and antioxidant level. So SINP and indole acetic acid were able to restore the chromium-induced arrested of cell cycle at G2 and phase. We have also analyzed some cell cycle analysis to take more clear observation of the impact of silicon and indole acetic acid. These are some results related to length, fresh mass, Si accumulation, and chromium accumulation. We have easily see that how silicon nanoparticle is more capable to reduce the accumulation, overall accumulation of the chromium accumulation. However, interestingly, when we apply silicon nanoparticle along with the indole acetic acid, we can see that significantly reduction, how these combinations significantly reduce the accumulation of the chromium accumulation in the plant cell. These are some other results related to the protein, chlorophyll, and carotenoids, and some photosynthetic pigments. These are the histochemical results which we have observed during our entire course of work. These are some histochemical parameters we have found. Now we should come to the main conclusion of this study. This is the overall mechanism that SINP and indole acetic acid inhibited chromium-induced oxidative damage, DNA damage, alteration of photosynthetic, and activation of antioxidant defense system. So what are the conclusion of this study is that the result manifested that combined application of silicon nanoparticle and indole acetic acid will be effective in alleviating chromium toxicity. Moreover, in SINP or indole acetic acid mediated mitigation of chromium toxicity, endogenous NO was shown to have a positive role. The study suggested that combination of silicon nanoparticle and indole acetic can be utilized against heavy metal stress to enhance the crop productivity parameters. So what are the main overall conclusion of this presentation, and what are the future perspective? So on the basis of overall discussion, we may convince that nanoparticle application can and will play a greater role in maximizing the global food production and protect the crop from the abiotic stress. Nanoparticle application may contribute to resolve the issues related to food demand of world population till 2050. So this is the overall impact of the nanoparticle which we have discussed. These are some differences which we have, and these are acknowledgement. These are some scientists all over the world which are associated with us, means Dr. Professor Jason C. White. He's a president from director from Chief Analytical Chemistry, Connecticut University. Professor J.S. Prelata, Professor Steven Shahi, Professor Rento Gario, Professor Francisco J. Carpus. These are some of our foreign collaborators. These are some of our Indian collaborators: Professor Sivesh Sharma, Dr. Devendra Kumar Chauhan, Dr. Vijay Pratap Singh, which is your another guest, a very eminent scientist, and he's one of my very best friend, and a very closest collaborators, you can say that. Professor Sriram Yadav from IIT Roorkee, and Dr. Rupesh Deshmukh Nabi Mohali. So these are my overall my talk. Thank you so much once again for the eminent scientist and the overall team of this local chapter for inviting me. I'm very much grateful to all of you. Thank you so much. If you have any question, you can ask.
Thank you, sir. If you have any question, you can write on chat box or unmute yourself and tell me. There is a physical audience. Student, do you have any questions? I can. Yes, come. Yeah, come here. Come.
Thank you so much, sir. As you told that there is a, like, if you can change the concentration of these nanoparticles, then they can influence the functioning in different dimensions. So my really question is, like, if a nanoparticle is induced, somehow it triggers free radicals, which leads to DNA damage or something like that. So my question is, like, how you want to deal with this problem as well? And also I had a very basic question, like, how these nanoparticles basically synthesize. Like,
Okay.
if we have,
Very good question. What is your name? Shivam. Okay, very nice question. So actually, the sequence of your question is a little bit not in a sequence. So first, your question is how we should synthesize the nanoparticle. Then how we can manage the DNA damage if due to the application of the nanoparticle, we analyze that it reduce the free radicals or any other thing. So first, there are many ways to synthesize the nanoparticle, right? Like we can utilize some chemicals to reduce the size of the nanoparticle, and the second is the green synthesis. We can extract the plant materials, and through this plant material we can synthesize through green synthesis technology. This is a protocol, a particular protocol. And third one is that we have discussed in that we can utilize the laser induced synthesis of the nanoparticle. This is the setup of the laser induced Bragdon spectroscopy. We can just we can utilize a pure rod of any metal, and we can just dip into the water, and then we can utilize the laser beam to synthesize the nano materials. Actually, the main thing is that if we will go through this synthesis of green material, then green synthesis method, then we can easily control the imbalance impact of the nanoparticle, the imbalance impact of the nanoparticle application, right? But if we will utilize the chemical synthesis, so the impact of these nanoparticle may be different, right, as compared to the green synthesis matter. Green synthesis method, right?
अच्छा लीव कर दे.
So hello, right? So Simam, so there are different methods of the synthesis of the nanoparticle. There are another method of the nanoparticle is ball milling, right? So we can also synthesize the one of the instrument, ball milling. Through the ball milling method, we can also synthesize the nanoparticle. So there are different chemical synthesis matter, chemical synthesis methodology, physical methods methodology, and laser induced synthesis of the nanoparticles. There are different methods, but one of the best methods is to synthesize the nanoparticle through the green synthesis, right? Like extraction of the plant extract, or as well as the utilize the laser induced breakdown spectroscopy or any other laser. Now the second question, answer of your second question is that if any nanoparticle shows the harmful impact or toxic impact into the plant system, how we can manage. So first, if you feel that any nanoparticle is damaging your plant, so you can just reduce their overall concentration, right? First, or you can apply any protective chemical agent like silica. You can use silicon dioxide, right, or nitric oxide, or any phytohormones. Then application of these protective chemical agent, you can control the toxicity of the any nanoparticle. If you feel that application of any nanoparticle is harmful for the plant system, or any physiological parameters, either it is related to biochemical parameters, or it is related to any molecular parameters, right? So you can control through this way. First, you should reduce the concentration. If you suppose if you are utilizing 100 micromole copper nanoparticle, and you find that this is the toxic, then you should go through the lower concentration of the nanoparticle. But it again depends on the size of the nanoparticle, that which size you are utilizing, right? Second, the nanoparticle you have synthesized, through which method it is utilized. It is synthesized by the green technology method, or it is synthesized by the chemical method, right? Because if you will compare the impact of chemically synthesized nanoparticle and the green synthesis nanoparticle, you will find a very significant difference between the both nanoparticle. if suppose you just you have synthesized copper nanoparticle through the utilization of some chemicals, as well as similarly you have utilized the copper nanoparticle, same copper nanoparticle through the utilization of plant extract. And then if you will apply into the any particular plant, like suppose rice or wheat, you will find that there are different concentration. Like if you utilize that 0, 5, 10, 50 and 100 micromole, you have two type of nanoparticle, chemically and green synthesized, and you just apply on the same concentration, you will find a significant difference between the both the nanoparticles, right? So it is depends on the method of synthesis of nanoparticle, as well as the as well as their size of the nanoparticle, right? So first you can control to reduce the overall concentration of the nanoparticle, and if it is not even, then if it is not possible, it shows the harmful impact as at the lower concentration, then you should utilize any protective chemical agent like nitric oxide, hydrogen sulfide, silicon, calcium, or any other protective chemical agent.
Okay. Thank you, sir.
Thank you so much.
Any other questions? Anybody want to ask? We have some very good. Dr. Akanksha Srivastava is saying to very informative and very motivational, motivation and informative lecture. And Satyendra Kumar Srivastava is also informative lecture. Nidhi Gupta, thanks. So from overall, it was really very nice lecture. Sir, thank you so much.
Thank you so much, Dr. Suren. Thank you so much.
Thank you very much, sir.
Thank you.
Okay, ma'am, over to you. Okay, sir, can you please stop sharing the screen?
Okay, yes. Yes, I'm stopped sharing.
Okay, now it's. Oh dear. Okay, so.
Thank you so much, sir, for your so many beneficial informations regarding the plants as well as our environment. Thank you so much, sir. Now, I would like to invite Dr. Sudhir Kumar Srivastava, sir, Treasurer ISCA, Dean of Science CSGM University and Director of IQAC DAV PG College, Kanpur, to propose a vote of thanks. Welcome, sir.
Thank you, ma'am. Honorable Dr. Vijay Lakshmi Saxena, she is connected online with us, President ISCA. Guest Speaker of the series, Dr. Vijay Pratap Singh and Dr. Durgesh Tripathi. Head of Department Botany, Dr. Jiten Mohan. Convener of this talk, Dr. Suren Pratap Singh, my colleagues, researchers, and dear students. It is a matter of pride for me that on behalf of the ISCA Kanpur chapter, I have been accorded the opportunity to stand before this august gathering of scientists and researchers to extend the vote of thanks. This lecture series is being organized in the name of legendary Dr. Nagen Swarup, whose vision for the education, administrative acumen, and capability as a mentor and guardian is well known to us. The lecture series organized is humble tribute to the memory of Karm Yogi Dr. Nagen Sarup. I convey my sincere gratitude to the Secretary Board of Management and the Council Member of Indian Science Congress Association, Mrs. Kumkum Sarup, for the inspiration and moral support for the event. I am delighted to express my profound thanks to Dr. Vijay Lakshmi Saxena, President ISCA, for great moral support and giving us valuable time to make this event successful. Her dynamic presence is always motivating for us. I am thankful to our guest speaker, Dr. Vijay Pratap Singh and Dr. Durgesh Tripathi, for their comprehensive, informative, and elaborate lecture. I am thankful for the valuable time given by them. I am also grateful to express my deepest thanks to Professor Arun Kumar Dikshit, Principal DAV College, Kanpur, for guiding us and providing perfect environment, which has been real motivation for all of us. I am thankful to Dr. Jiten Mohan for his support. I thank all the distinguished invitees present here, online and offline, accepting our invitation, and all good hearts who work behind the screen. I am also thankful to convener Dr. Surendra Pratap Singh. He has been very diligent, energetic, and innovative in this entire process. I am also thankful to Dr. Charu Chaturvedi for her decent comparing of the event. I owe special gratitude to all who have worked hard to ensure that this occasion becomes a memorable success. Once again, I thank you for all your attention. Thank you so much.
Thank you so much, sir. Please stand for the national anthem. Now it's time for tea. Please.
So everyone
come join
invited
us.
for a tea and snacks who are there in MSc previous. So thank you all. Sudhir sir, allow me to close this session. Okay. Thank you all. Thank you. Thank you for the all. Okay. So Swami, thank you for organizing such a wonderful. Thank you so much. Thank you, everyone. Thank you. Thank you. So we will organize in the next lecture very soon. Thank you.